Hi all, welcome to my new series of video lectures. Today we are going to see about how we came to know the DNA as a genetic material. Since DNA is discovered so long back, but it took some years for scientists to convince themselves as a DNA as the genetic material. They need some convincing proof of experiments to get convinced that DNA is the only genetic material. So many scientists who worked on that, many experiments they did. At last one day they came to know that, or they were convinced that DNA is the genetic material. Today we are going to see that what are all those experiments and who are all those people who are involved in that work. Before going on to the topic, let us see these basic things about the chemical nature of the gene. Gene is nothing but DNA. Early scientists thought that protein was the genetic material, and DNA was thought to be repetitious and have little complexity. As we know that proteins are made up of amino acids, and we have nearly 20 essential amino acids which are present in the protein. And DNA, when it comes to DNA, it has only 4 bases, and it was keep on repeating, and they are not convinced that it is going to be the genetic material. So not enough complexity to be the genetic information. And proteins were complex, they are variable. So this complexity suggested that the proteins could be the genetic information. So we'll see a little bit on the history on the description of the structure of DNA. It was started long back in 1869. Fritz Meischer discovered that the nuclei of pus cells contain an acidic substance, to which he gave the name nuclein, as we call the nucleic acid. So we know that they are acidic by nature, and Fritz Meischer is the first one who identified that in the nuclei of pus cells. Later he find that nuclein is composed of a protein and a compound to which the name nucleic acid and subsequently DNA will be given. In 1928, Frederick Griffith, we are all familiar with him, finds that a substance in a heat kill bacteria can cause heritable changes in the live bacteria alongside them. He calls the phenomenon called transformation. In 1944, Oswald Avery, Colin MacLeod, and Maclean McCarthy established the chemical identity of Griffith's transforming principle as DNA, and they suggest that it may function as the genetic material. Actually, these 3 people are taking the experiment of Griffith into the next level. And in 1949, Erwin Chargaff reports that the DNA-based composition varies from one species to another. Yet the ratio between the quantities of the 2 purine bases, adenine and thymine, and between the quantities of the 2 pyrimidine bases, guanine and cytosine, remains about the same, namely 1 to 1. That's one of the greatest achievements. In 1951, Rosalind Franklin distinguishes 2 forms of DNA, the paracrystalline B form and the crystalline A form. 1952, Hershey and Martha Chase find the DNA, but scarcely. Just a minute. In 1952, Hershey and Chase find that DNA, but scarcely any protein from an infecting bacterial virus enters a bacterial cell and can be recovered from the progeny virus particles. In 1952, Rosalind Franklin and Raymond Gosling produced a magnificent X-ray diffraction pattern of B form of DNA. In 1953, James Watson, which is a biggest milestone. 1953, James Watson and Francis Crick, Rosalind Franklin and Raymond Gosling, and Morris Wilson and W.E. Seeds, Alex Stokes and Herbert Wilson and Bertil Jacobson all published on the structure of DNA. In 1957, Francis Crick proposed the sequence hypothesis on the central dogma. In 1958, Matthew Michelson and Franklin Stahl demonstrated the semi-conservative replication of DNA. In 1951, Arthur Kornberg and his colleagues isolated the enzyme called DNA polymerase. 1961, Marshall Nierenberg and Jonathan Heinrich Mathai showed that the sequence of nucleotide can encode a particular amino acid, laying the foundation for deciphering the genetic code. 1960, the Nobel Prize in Medicine awarded to James Watson, Francis Crick, and Maurice Wilkins. These were the few achievements that were made in this path. Let's go to the topic straight away. And our topic is the evidence for DNA as a genetic information. As I said before, so many people works on this to prove that DNA is the only genetic material. And the first and the pioneer among them was Griffith. In 1928, he was working with a pathogenic strain of pneumococcus. As we all know about this pneumococcus, it is a bacteria that causes fatal pneumonia in animals. In that he took two strains. One is called S strain and another one is called R strain. S strain is called smooth strains and are pathogenic. R strain are called rough strains and they are not pathogenic. So this is what he took. He took a S strain and he took a R strain. When he injected, as I said before, S strain is pathogenic, R strain is non-pathogenic. When he injected the S strain, the mouse died. The living S strain cells isolated from the heart. And when he injected the living R strain, which is non-pathogenic, and the mouse is healthy, and no bacterial cells is found in the heart. And then he did a heat killing. He did a heat killing of this pathogenic S strain, and he injected into the mouse. It was healthy. It has not died. So no bacterial cells found in the heart. But when he mixed that heat killed S strain and then non-pathogenic R strain, the mouse dies, and he isolated living S strains from the heart. So he concluded like this: a chemical component from one cell is capable of genetically transforming the another. So what he's trying to tell is, here the last one, he used a heat killed S strain. It means that is inactive, and a living R strain, just non-pathogenic. So something has happened that transformed the non-pathogenic R strain to a pathogenic, to become a pathogenic. Okay, so this is what. So he concluded like this: a chemical component from one cell is capable of genetically transforming the another cell. So that was his conclusion. Something is transforming. Then there are another three persons, Avery, MacLeod and McCarthy. They took this into the next step. So he tested various extracts of S type, the same pathogenic strain of pneumococcus. So they extracted nucleic acid, proton, insoluble, etc. The only nucleic acid fraction transformed the R type cells. Only the nucleic acid fraction transformed the R type cells. The DNA treatment eliminated the transformation. So we'll see it a little more briefer through this image. So they took a heat killed S cells, okay, and then they made 3 aliquots from this. Okay, in one aliquot they added proteinases. As you know, proteinases are enzyme that degrades protein. They added RNases. RNases are enzyme that degrade RNase, and DNases are enzyme that degrade DNA. To that he added R cells. R cells are non-pathogenic. So you know that. And wherever this proteases he added, in that one transformation occurred. And wherever this RNases he added, transformation occurs. Wherever he added DNases, the transformation doesn't occur. So no transformation occurred. So what he concluded was, the transformation cannot occur unless DNA is present. This is one of the major conclusions he did. The transformation cannot occur unless DNA is present. Therefore, DNA must be a hereditary material. Yes, they have taken the steps. They have taken this research into the next step. But still, though, it was not convincing for most of the scientists. they didn't get convinced that DNA is genetic material still. So the other two people are coming into the game. They are called Hershey and Chase. Here they are using a phage. Previously the two group of people, they used bacteria. Here is using a bacteriophage which infects E. coli, and the strain they used is T2. So T2 is a phage which infects E. coli. So this T2 is well studied. Even in 1952 itself, they have little knowledge about this phage. It was already known as T2 protein contains the elements S but not P. So we know that the protein contains sulfur but not phosphate. But DNA was not known to have lots of phosphate but not sulfur. And DNA protein can be distinguished on the basis of this elemental analysis. So what this Hershey and Chase did was they labeled radioactively. Okay. So Hershey and Chase grew T2 in the presence of radioactive S35 and radioactive P32. Got T2 labeled with S35 protein and T2 labeled with P32 DNA. As you know that, as I said before, sulfur is present in a protein. So sulfur will go and bind automatically to the protein, and phosphate will go and bind automatically to the DNA. Since they are radioactively labeled. And infect E. coli with the label T2 phage and see where the radioactivity goes. As you know that this bacteriophage will infect the host by inserting its DNA into the host and not its protein. So protein is present in its shell and it injects its DNA into the injects its genetic material into the host. The virus T2 bacteriophage, this is what I said. So it has a head, neck, collar, sheath, so many parts it has, and the DNA is present here. So the and it gets injected into the E. coli, which is the host. So the first experiment, they took a 32P radioactively labeled containing DNA. Actually at this time they don't know that they took a 32P containing DNA and they infected the bacteria. They spinned it. As you know that, so this bacteriophage will insert its DNA into the host, and the remaining particle is called a ghost particle, and it will be removed on centrifugation. After removing, if you see, after the centrifugation, they got very little 32P in the supernatant, because this phage particles are ghost particles. It is present in the supernatant, but during the infection, this 32P get transfected into the host, so it went and settled down. So at the supernatant, they found very little 32 phosphorus. The experiment 235S containing phage codes used, the same thing happened, but here the sulfur will go and bind to the protein, not to DNA. So after centrifugation, whatever they did, so they found nothing in the pellet. The remaining, the ghost particle, which has a protein in which this 35S sulfur is attached, it is present in the supernatant. So from the supernatant, they found most of the 35S particles. So conclusion, DNA, not protein, enters the bacterial cells and directs the assembly of the new virus particles. This is one of the ultimate conclusion they made, and it was very convincing among the scientists to consider that DNA is the genetic material and not protein. And this is the original article they published in the year 1943. I hope this lecture was useful for you. Please like this video and please post your valuable comments so that it will be helpful for me to improve a lot. This is just an initiative and I have a lot of plans to do it. I hope you support me.